So welcome to mitosis. You're already very familiar, hopefully, with the parts of a cell. The parts of the cell that we focused on were those organelles that were directly involved in reproduction. And as you know, cells are not dead. So assuming that they're not dead, they actually have to repair themselves, they have to grow, and in the case of some organisms, they actually reproduce by mitosis. So mitosis is primarily that type of cell division where your body cells are replacing cells constantly, and you need to know the names of the steps involved and be able to recognize just what is happening in a nucleus when the cell divides. So lots of resources for this. Rather than me specifying particularly which ones you have to do, if you take a look at the log file, I have put down a number of suggestions. The piece of paper you're looking at right here, of course, is not an option because it's basically the place to start. I would start with going through some of these mitosis step by step, the cell cycle, cytoplasmic division, and brain pop. And then in this actual podcast, BobbyTalks Mitosis, if you stay with it, there's a whole bunch of notes, there's a PowerPoint, and a whole lot of suggestions as to just what you should be writing down in this little box. Some of the resources are great, some of them are not so great. Some of them you'll like, some of them you probably could care less. And you know what? There's more there than you need. So once you know the cell cycle and the divisions within mitosis and can actually recognize those, then you know what? Feel free to stop. So make sure you read this, and yeah, we'll move on to the cell cycle. Just a quick reminder of where the best resources are for actually mastering mitosis. And again, like I said, other than that one particular handout that I just showed you, the options here are ample, right? So yeah, my favorite, quite honestly, is this one, Mitosis Like You've Never Heard It Before. I believe that's our friend Hank from Crash Course Biology. Really good. If you want just the essentials, Bozeman Science Mitosis, if you're on a nice device that has app capability, download the Mitosis app. Again, everything you could possibly ever want to know about mitosis. There's going to be a few labs here where you have an option. I'm quite happy if you do any two of these. Cheek cell lab for real, your own cheek, or a virtual option. Mitosis onion root lab, again, virtual. Mitosis internet lesson, and there's probably even a couple more I'll put up there. So a lot of different ways of learning this. Just make sure you record it in your log file as to what you actually did in order to learn about mitosis. So let's first consider the cell cycle. You are alive. You are made of cells. Your cells are not just staying in one particular stage of their life cycle. Let's call this the cell cycle. I'm actually going to label it here for you. I think I'm going to label it here for you, but for some reason or other, when I use my sweet pen, the entire diagram moves, so that is not good. So let's try this again. Cell cycle. If anybody knows how to rest one hand on a screen and at the same time write on that screen without having the image move, I would appreciate a little lesson in it. So this is the cell cycle. It's just a diagram representing the fact that the cell is actually going through stages where it is dividing. That is called the mitotic phase. Mitotic phase. And that's really what this chapter is about, the two parts of the mitotic phase. One, of course, is mitosis. That was an M, honest it was. And following mitosis and all the lovely stages in mitosis, we have something called cytokinesis. Cytokinesis. Don't be afraid of words like cytokinesis. Cyto just means cell. Kinesis means splitting or movement. Get the word kinetic energy, energy of movement, or cinema, moving pictures, from cytokinesis. So that is the stage, the mitotic phase, where a cell is dividing. One cell becomes two. The rest of the time, which is most of the time, the cell is in a cycle called interphase. Okay, 3 stages of interphase. One is called G1, which just means growth. The other one is called S for synthesis. That's where protein synthesis takes place. And the next one is called G2, where again the cell is growing. So that is the cell cycle. Quite honestly, this section of your chapter is really focusing on mitosis and those stages. So we'll move on to that. So the stages of cell division, in particular, this is stages of mitosis. You should consider that a cell starts here, number 1, as a single cell, and then this of course shows the nucleus with all the genetic material in the middle, the chromosomes made up of DNA. It goes through a number of stages, and the very first place that we start is with something called interphase. Inter just means between, stages. So this is where we start, and it's also where we end up, back at interphase. So that's where a cell spends most of its cell life, not actually going through mitosis. But when it does begin to divide, things happen in the nucleus, and you're noticing that the nuclear membrane is beginning to break down, and you're actually beginning to see the chromosomes come together. So we'll go into more detail as to what actually happens in each of these phases in the little PowerPoint that follows, but for now realize that the second stage is called prophase. And following prophase, we have number 3, which we will call metaphase. Quite honestly, this is the one that is usually the easiest to distinguish, because you'll see these chromosomes are actually in pairs lined up towards the equator, or the middle of the cell. Then, in the 4th stage, which we call anaphase, those chromosomes begin to split into centromere going one way and one the other way. So you now have chromatids that are dividing at the centromere and being pulled towards the spindle. This is a spindle on one side of the cell, spindle on the other side of the cell. And then finally, the last stage, number 5, what we call telophase. Telophase. You actually begin to see the formation of 2 new cells pinching off, and the nuclear membrane being reformed. Okay, looking at the cell cycle diagram just in a slightly different way, diagram from your textbook, just in case this confuses you. Cells spend most of their time in interphase. I think we've mentioned that one already. Growing and performing their specific functions. Cell division includes both mitosis and the cell splitting itself, called cytokinesis. So if you look at the cell cycle, notice that most of it is in interphase, but those cycles we identified as G1 and G3 are here. Duplication of chromosomes happens in the S phase, and then this cell division is, of course, mitosis and cytokinesis. So not to worry at all. Stages of mitosis, and again, these are the stages you need to be able to recognize: prophase, metaphase, anaphase, and telophase or telophase. So we start off with a cell in interphase. You notice the nuclear membrane breaking down, the actual formation of the chromosomes in a recognizable form, and then in metaphase they pair up and basically align themselves to the middle of the cell along the cell equator. Anaphase, they split into separate chromatids and pull to the opposite poles. And finally, in telophase, the cell membranes pinch in, and you end up with the end product, 2 new daughter cells. So cytokinesis is the process of dividing the cytoplasm in 2, and occurs in different ways in animal and plant cells. The products of cell division are two genetically identical cells. So these two cells here are identical genetically to the one you started with. That is the cell cycle. Okay, let's look at this wonderful mitosis thing in its most simplified way we possibly can. Remember, the stages are prophase, metaphase, anaphase, telophase, and they are bracketed by interphase on both sides. So the way I always have remembered this is we were doing this one day in class when the Prime Minister happened to be in India, so I suddenly, in my brain, it says Prime Minister at India. Prophase, metaphase, anaphase, telophase in India, interphase. So prophase basically is where replicated DNA undergoes multiple spiraling and becomes visible under the, light microscope as chromosomes. Spindle microtubules start to form. These are the structures that the chromatids slide along towards the poles of the cell, and then the nuclear envelope itself begins to break down. So you, at this stage, at the end of it, you probably wouldn't even see the nucleus. So if I was going to draw it, I would draw a chromosome, two chromatids. This circle in the middle is the centromere. These are the spindles at either end, and let's move on. So metaphase is where chromosomes move to the central plate or the equator of the cell. The microtubule network is completed. They attach the centromeres, and you would end up with something like this. So now we're along the equator of the cell. Notice that the nuclear membrane is no longer visible. So sometimes you will be asked to actually draw a stage of mitosis. So the reason we're showing it in this very simplified manner is that that's probably how I would draw it. I'm not an artist, so we're picking a cell here that only has two chromosomes. Obviously, much higher organisms have many more chromosomes than two, but this is a simpler way of looking at it. So following metaphase, we have anaphase. Centromeres break, the microtubules contract, and with them the sister chromatids get pulled to opposite poles. So there is what is happening in that stage. And what do you think comes next after anaphase? Telophase. Telophase, the chromosomes uncoil, and of course as they uncoil they're less dense, they're less visible. Under a light microscope you would probably not even see them, and the spindle microtubules themselves break down, nuclear envelope reforms, and you would have two cells that are identical to the one original cell genetically. Mitosis.
All living organisms are composed of cells. Organisms composed of single cells are unicellular, whereas multicellular organisms are composed of many cells. You are a multicellular organism. Your body is made up of trillions of cells. And yet at conception, you are no more than a single cell formed from the union of a sperm and an egg. In this program, you will learn how unicellular organisms, known as prokaryotes, divide through binary fission, and how non-reproductive cells of organisms known as eukaryotes divide through mitosis and cytokinesis. Before learning how cells divide, it's important to understand how cells arise. All cells come from other cells. From that single cell at conception, you developed into your current size and proportions through repeated cell divisions. Even now, your cells are continually dividing to replace dead or damaged cells. In fact, cell division in a healthy adult human occurs at the rate of around 25 million new cells produced every second. In unicellular organisms, such as bacteria or these tiny diatoms, cell divisions create new individuals. In multicellular organisms, such as horses or humans, cell divisions result in growth or replacement of cells. It's important to realize that an organism grows because its cells divide to make more cells. Although cells often grow some after they divide, growth of an organism does not occur simply through the enlargement of its cells. If cells continually grew, they would reach a size where transporting factors necessary for survival into and out of the cell would be hindered, and growth would come to a standstill. The cells of a baby penguin are the same size as those of an adult penguin. The reason the adult is larger is simply because it has more cells. You've learned that all organisms are made of cells, and that new unicellular organisms and growth of multicellular organisms result from cell divisions. We will now examine the processes of cell division in the two types of cells found in different kinds of organisms: prokaryotic cells and eukaryotic cells. Bacteria and blue-green algae, also known as cyanobacteria, are unicellular prokaryotes, whereas protists, plants, fungi, and animals are composed of one or more eukaryotic cells. All cells, regardless of whether they are prokaryotic or eukaryotic, carry information in the form of deoxyribonucleic acid, or DNA. DNA tells cells how to build the molecules and structures necessary for survival, as well as how to divide into new cells. In both types of cells, DNA is wrapped around proteins and condensed into long thread-like fibers called chromatin. The chromatin can be further condensed into structures called chromosomes, which we will learn about shortly. Sections of DNA in the chromatin or chromosomes code for specific traits and are known as genes. For example, genes code for the traits of hair color in humans and the structures that allow plant cells to perform photosynthesis. There are even genes for making molecules that control when and how a cell divides. Cell division in both prokaryotic and eukaryotic cells involves a duplication of chromosomes so that each new cell will have the genes it needs to survive. A major difference between prokaryotic and eukaryotic cells is the manner in which DNA is housed in the cell. In prokaryotic cells, the DNA typically exists in a single circular chromosome. Prokaryotic chromosomes are found in an area called the nucleoid region, which is not separated from the rest of the cell contents by a membrane. The electron microscope image you see shows part of the circular chromosome that was isolated from a bacterial cell. In contrast, the DNA of eukaryotic cells is packaged into a number of chromosomes, which varies from species to species. For instance, typical human cells have 46 chromosomes, whereas cow cells have 60 chromosomes. Chromosomes are located within a structure called the nucleus. A double membrane called the nuclear envelope separates the contents of the nucleus from the rest of the cell. The entire area between the nucleus and the cell membrane is called the cytoplasm. Membrane-bound organelles with specialized structures and functions reside within the cytoplasm. In this microscope photo of eukaryotic cells, the structure in the center of each cell is the nucleus. The cytoplasm is the fluid-like substance surrounding the nucleus. The dark region within each nucleus is called the nucleolus, a site where structures used in making proteins are being produced from specific genes in the DNA. Note that you cannot see individual chromosomes. These structures are only visible through the light microscope just before the cell starts to divide. The method of cellular reproduction differs between prokaryotic cells and eukaryotic cells. Prokaryotic cells reproduce by a type of cell division called binary fission. Binary fission, shown here in bacteria, starts with the replication of the chromosome. Next, a portion of each the original and duplicate chromosome attaches to the plasma membrane of the cell. The cell nearly doubles in size and then pinches in half, dividing the original parent cell into two new daughter cells, each with identical copies of the DNA that was in the parent cell. Under optimal environmental conditions, bacterial cells can mature and divide by binary fission in as little as 20 minutes. On the other hand, the cells of higher eukaryotes, like humans, typically take from 16 to 24 hours to mature, replicate their chromosomes, and divide. The difference in time between prokaryotic and eukaryotic cell division reflects to some degree the complexity of the cells involved. Eukaryotes have a larger number of chromosomes to duplicate, and they have a nucleus and membrane-bound organelles that are not present in prokaryotic cells. The process in which eukaryotic cells duplicate their chromosomes and distribute them to new cells is known as mitosis. The process in which the contents of the cytoplasm are divided into new daughter cells is called cytokinesis. Although your eukaryotic cells average around 20 hours to mature and divide into daughter cells, they differ in the length of this cycle almost as much as they differ in the jobs they perform for your body. For instance, skin and intestinal cells, which are worn off or destroyed daily, continuously divide. Nerve and muscle cells, such as those found in the brain and the heart, rarely, if ever, divide once they are created. Plants undergo rapid cell division near the tips of the roots and stems, but division is much less frequent in other areas. The cells of an early animal embryo divide at an astonishing rate. They divide in as little as 30 minutes, but they do not spend much time growing because they are primarily dividing up cytoplasm that was present in the egg cell at conception. Due to different cell maturation and division rates, it is most convenient to discuss the processes of cell growth and division in eukaryotic cells in terms of their cell cycles. Eukaryotic cells go through a distinctive cell cycle in which they grow, replicate the chromosomes, and prepare for cell division before undergoing mitosis and cytokinesis. The chart you're seeing illustrates the different phases of the cell cycle. The G1, S, and G2 phases make up what is known as interphase. During interphase, the cell grows, produces more cell structures, and doubles its chromosome set. Eukaryotic cells spend the majority of the cell cycle in the G1 phase of interphase. During G1, or the first growth phase, a cell grows, makes new cellular molecules and components, and performs the normal cellular reactions necessary for survival. Cells in your stomach would be producing digestive enzymes to help break down food you ate at your last meal. Skin cells would be maintaining themselves while exchanging waste materials produced through cell metabolism for nutrients from the blood and capillaries. Plant root cells would be taking in nutrients or water from the soil, and leaf cells might be photosynthesizing. During the S, or synthesis phase, the DNA in the nucleus is duplicated to form identical copies of each chromosome. Each new portion of the duplicated chromosome remains attached to the original by a highly compacted region of DNA known as the centromere. The individual members of the attached pairs are called chromatids. The chromatids of each chromosome are commonly referred to as paired or sister chromatids. However, at this point of the cell cycle, the chromosomes are not singly visible because they're still in the form of chromatin fibers. In each cell, protein fibers called microtubules form a network called the cytoskeleton that maintains the shape of the cell. Another important event occurs during the S phase. Microtubules also accumulate and organize at a region near the nucleus, known as the centrosome. In animal cells, some of the microtubules also form a pair of compact structures called centrioles. During the S phase, the centriole pair separates and they are duplicated. Plant cells have no centrioles, but they do have microtubules that are important in mitosis. In the last phase of interphase, G2, or the second growth phase, the cell continues to grow, creates new organelles, and metabolizes normally as it readies itself for the M phase. The centrosomes begin to separate, and compounds that control entry into mitosis, the first part of the M phase, build up. These compounds are generically called cyclins and MPF, which stands for maturation, or M phase promoting factor. MPF concentrations begin to rise during the S phase, and when enough MPF builds up during the G2 phase, the cell enters the M phase of the cell cycle. The M phase consists of mitosis and cytokinesis. This process is relatively rapid in most eukaryotic cells and generally takes about an hour. Mitosis consists of 4 stages: Prophase, Metaphase, Anaphase, and Telophase, during which time the cell's paired chromatids divide in half and move to opposite sides of the cell. Mitosis is followed by cytokinesis, the process in which the cytoplasm of the parent cell is separated into 2 equal portions, forming the daughter cells. Of the 4 different stages of mitosis, the first stage, prophase, is the longest. Prophase accounts for around 50% of the time spent in mitosis. As cells enter prophase, their chromosomes are doubled, but they are still long, indistinguishable chromatin fibers. During the first part of prophase, however, the chromatin condenses to become the individually visible chromosomes. In fact, the term mitosis stems from the Greek word mitos, which means thread, and refers to the condensed visible chromosomes. Inside the cytoplasm, the centrosomes separate and begin to move toward opposite sides or poles of the cell. These star-shaped bodies, which are comprised of microtubules, and the centrioles in animal cells, are known as asters. Initially in prophase, the nucleus is very prominent and contains a distinguishable nucleolus. As prophase progresses, however, the nucleolus and nuclear envelope enclosing the nucleus slowly disappear. The nuclear envelope fragments into small sac-like vesicles that remain present in the cell. This photograph of animal cells in prophase shows the condensed chromosomes and the lack of a nucleus and nucleolus. From the asters that have migrated toward the poles and their accompanying centriole pairs in animal cells, fiber-like microtubules extend and begin to form what is known as the mitotic spindle. Some of the microtubules comprising the spindle fibers attach to the centromeres that hold sister chromatids together. Other spindle fibers stretch across the middle of the cell and start to shape the mitotic spindle. Attachment to microtubules sets the chromosomes in motion, and they begin to travel toward the equator of the cell, which is called the metaphase plate. Some biologists draw a distinction between this stage of prophase and the beginning of prophase, in which the chromosomes condense. Those who draw this distinction refer to this step as prometaphase. That is, it's referred to as an intermediate step between prophase and the next phase, metaphase. During the phase of mitosis, known as metaphase, the spindle is completed and the chromosomes line up along the metaphase plate. Metaphase is the shortest phase of mitosis and often lasts only a few minutes. These photographs show plant and animal cells undergoing metaphase. Plant cells lack centrioles, but microtubules still form a spindle and enable the chromosome to align at the metaphase plate. The third major stage in mitosis is known as anaphase. Like metaphase, the process of anaphase is short and typically occurs in a matter of minutes. During anaphase, centromeres holding sister chromatids of each chromosome together separate. At this point, what were once known as sister chromatids of the same chromosome are now recognized as two identical individual chromosomes. Each chromosome is then pulled away from its prior partner, and they travel toward opposite poles of the cell. Chromosome migration toward the poles occurs at a steady pace and results from two processes. First, microtubules attached to the recently separated centromere region shorten and pull the chromosomes toward the poles. Second, the microtubules that form the rest of the mitotic spindle extend and begin to elongate the cell, thereby further separating the chromosomes. Eventually, each side of the cell receives one complete set of chromosomes. These photos of plant and animal cells near the end of anaphase clearly show the chromosomes. Notice that they have separated and migrated toward the poles of the cells. Anaphase is followed by the final stage of mitosis, telophase. In telophase, the chromosomes have migrated to opposite poles of the cell, and the microtubules they were attached to break up and disappear. The microtubules that form the spindle continue to extend and further elongate the cell. Vesicles containing fragments of the original nuclear envelope, which disappeared during prophase, fuse and begin developing into new nuclear membranes. As new nuclei start to arise around each set of chromosomes, the chromosomes unravel to their less condensed chromatin form. Eventually, the new nuclei are completed and new nucleoli appear. The process of mitosis, the equal division of the genetic material in the nucleus, is now complete. This photograph shows a plant cell in telophase. Notice that there are two distinct nuclei, and the chromatin can no longer be distinguished as individual chromosomes. The final process of cell division, in which the cytoplasm splits and the two new nuclei, with their identical genetic material, are distributed to daughter cells, is called cytokinesis. Cytokinesis actually starts around late Anaphase, but it is not readily observable until the end of Telophase. Earlier in the program, you learned that MPF, maturation or M phase promoting factor, builds up at the end of the G2 phase of interphase and triggers the cell to enter mitosis. MPF continues to build until the cell reaches metaphase, where it levels off. In anaphase, MPF starts to break down. This decrease in MPF signals the cell to finish the final phases of mitosis and begin the process of cytokinesis. The concentration of MPF continues to rise and fall with each turn of the cell cycle. Unlike mitosis, which is fairly similar between plant and animal cells, cytokinesis differs markedly between the two cell types. Let's first examine cytokinesis in animal cells, which is also known as cleavage. In most animal cells, spindle microtubules that remain near the cell equator are surrounded by a ring of contractile filaments, comprised of the proteins actin and myosin. Actin and myosin, as you may already know, are proteins responsible for muscle contraction. In cell division, the ring of contractile proteins they form is called the contractile ring. The contractile ring attaches to the plasma membrane and pulls it inward, creating a depression around the cell called the cleavage furrow. Once formed, the contractile ring continually loses filaments, causing the cleavage furrow to deepen. Eventually, the contractile ring surrounds the narrow ring of microtubules remaining from the mitotic spindle. This narrow bridge, called the midbody, eventually breaks and cleavage is complete. The cell has essentially pinched in half, forming two new daughter cells. Plant cells undergo a different form of cytokinesis, mainly because their rigid cell wall is not capable of pinching inward, like the cell membrane of animal cells. Instead, plant cells divide by a process called cell plate formation. Vesicles derived from the Golgi apparatus carry cell wall precursors to the equator of the cell, along the microtubules remaining from the mitotic spindle. There, the vesicles fuse to form a disc-like structure known as the cell plate. The cell plate is a double membrane-bound structure that expands outward as more and more vesicles carrying cell wall precursors fuse. Eventually, the cell plate reaches the existing plasma membrane, where actin proteins direct its attachment. The cell is now essentially divided into two halves. Cell walls then develop between the layers of the cell plate, completing the division of the parent cell into two new daughter cells. The photographs you see show plant and animal cells just after they have completed mitosis and cytokinesis. As you can see, cytokinesis typically divides the cytoplasm so that each of the two daughter cells receives an equal share. The cellular organelles, which were duplicated or partitioned into vesicles before mitosis, eventually end up in each of the daughter cells before cytokinesis is complete. Once cytokinesis is complete, the cell cycle starts anew. The daughter cells enter the G1 phase of interphase, and the organelles begin to grow and perform the functions necessary for cell growth and survival. This concludes our look at cell division and mitosis. You've observed the process of binary fission in prokaryotic cells, as well as the eukaryotic cell cycle and the processes of mitosis and cytokinesis. You now know how organisms, such as bacteria, create entirely new organisms, and how cell division allows multicellular organisms to grow and replace dead and damaged cells. The next time you look at an organism, either through the microscope or in your own backyard, you'll be able to appreciate these vital cellular processes.